Good day. We are the groups 1 and 2 of Section 1 and we are here to present our mock methodology about transcription factors of cell growth.
For our objectives, to determine and apply different molecular techniques and its principles and methodology for the transcription factors of cell growth.
I'm Carl and I'm here to give a brief introduction about transcription factors. So let's define what is transcription. Transcription is the process where a gene's DNA sequence is copied into an RNA molecule. On the other hand, transcription factors are proteins that help specific genes on or off by binding to nearby DNA. In short, they are like the regulators. So transcription factors has two kinds. First one is the general transcription factors which are required for transcription initiation, and the last one is specific transcription factors which increases transcription in certain cells or in response to signals.
Our mock methodology includes number 1, reagent preparation, 2, DNA extractions, 3, nanodrop testing, 4, gene amplification via RT-PCR, 5, gel electrophoresis, 6, analysis of results. First, we're going to tackle about the reagent preparation. Reagent preparation. The reagents listed below are the most commonly used when it comes to transcription factors of cell growth. It is based on the journals that is used in this presentation. For the reagent preparation, the reagents used are 3.5 ml saturated culture into 100 ml of fresh MSS media or the Murashige and Skog salt, 3% weight over volume sucrose, 0.5 mg per liter NAA, 0.05 mg per liter kinetin with pH 5.8 using 1 NKOH, triazole kit, extracted RNA with RNase 3 DNase 1 sigma, 1% weight over volume formaldehyde agarose gel before and after DNase 1 treatment, 2.5 microliter of SYBR Green Master Mix, superscript transcriptase kit in Vitrogen, 5 microliter contains 0.5 microliter of template cDNA or RNA, 2 microliter of mixed 0.5 mm forward and reverse primers.
For the transcription factors of cell growth, we will be needing the following equipments: Neubauer counting chamber, Olympus BX51 microscope, Erlenmeyer flask covered with aluminum foil, spectrophotometer, real-time polymerase chain reaction, and Abbey Prism 7900 HT sequence.
Next is DNA extraction. In the DNA extraction needed for identification of transcription factors of cell growth, the journal cited in this presentation used the Fecal Plate Plus. Fecal Pack Plus. Cells were cultured with stem cell medium in dishes coated with an ultra-low attachment surface for 1 week. Refeed with SCM every day. Load onto fecal media, centrifuge for 30 minutes at 1080 RPM with zero deceleration. Collect interphase layer, wash with phosphate buffered saline. Enriched cells are now ready to use.
Now let's proceed to the third part of our presentation, which is all about nanodrop testing.
For nanodrop testing, first, each source sample RNA was labeled with CY3 dye as per the protocol detailed in the low input quick amp labeling kit. Second, A specific activity of greater than 6.0 was confirmed by measurement of 260 nanometer and 550 nanometer wavelengths with a nanodrop ND1000 spectrophotometer. Third, labeled RNA was hybridized for 16 hours to Agilent SurePrint G3 8x60K microarray slides. And fourth, after hybridization, slides were washed as per protocol and scanned with a high-resolution CE scanner using a scan resolution of 3 micrometer. Lastly, feature extraction was performed using Agilent feature extraction software with no background subtraction.
To continue, let us now proceed to our part 4, which is all about gene amplification, real-time PCR, or polymerase chain reaction.
Total RNA was isolated using Prizol reagent. PCR was performed using a 25 microliter system with hot start DNA polymerase. Complementary DNA or cDNA was synthesized by a thermoscript reverse transcription PCR system according to the manufacturer's instruction. The PCR products were separated on a 1% agarose gel.
The PCR used for the amplification method is the reverse transcription PCR using the hot start DNA polymerase for 36 cycles. The initial incubation of the cycle should be 95 degrees Celsius for 30 minutes. The melting or denaturation occurs in 94 degrees Celsius for 30 seconds. After denaturation, the temperature will cool down in a 60 degrees Celsius temperature. Then after, annealing will occur in 53 degrees Celsius for 30 seconds. Lastly, the extension phase, which occurs in high temperature, 72 degrees for 60 minutes, and then the 72 degrees for 10 minutes at the last cycle. The PCR products will then separated on a 1% agarose gel.
Next, for our mock methodology is the number 5, which is gel electrophoresis. Gel electrophoresis.
For gel electrophoresis, the total cellular protein was extracted using the RIPA buffer and then quantified using ABCA kit. Next, lysates containing 50 microliters of protein in each sample were analyzed by SDS page. The proteins were then transferred to a polyvinylidene difluoride membrane, and residual protein sites were incubated with different primary antibodies, followed by the appropriate horseradish peroxidase conjugated secondary antibody for protein visualization with ECL reagents.
Down to our last part for our mock methodology is the analysis of results. Analysis of results.
For the analysis of results, SP1 was the first identified and is now one of the best mammalian transcription factors studied. It serves as a transcriptional activator of a number of genes, including genes for housekeeping, regulators of cell cycles, and genes restricted to tissues. In this study, SP1 was used for early embryonic development but dispensable for cell growth and differentiation by targeting the inactivation of the mouse gene encoding transcription SP1.
In this slide, you can see the figure represents the results for the targeted disruption of the SP1 gene. For the first figure A, two exons encoding the three zinc fingers and the C-terminus of SP1 are replaced. Figure B is a Southern blot analysis of the targeted ES cells. Probe A detects 5.6 kb band, a wild-type allele A, or 9.0 kb band for the targeted allele.
For probe B, it detects a 9.2 kb band A wild-type allele or 8.6 kb band, the targeted allele. For the figure C, the PCR analysis of the moist embryos, wherein the primers detected a 378 pp band for wild-type allele or the 690 pp for the targeted allele. Thus, SP1 may be linked to the cell cycle and the making of transcriptional competence by maintaining methylation-free islands of the chromatin structure.
Here are the 5 journal references that we use in mock methodology for the transcription factors of cell growth.